A drug that has been prescribed for high triglycerides for decades has just produced a completely unexpected result in aging research. Scientists found that gemfibrozil improved healthy lifespan across multiple experimental species, and the mechanism did not appear to depend on the pathway doctors normally associate with this drug. Instead, the researchers traced the effect to a nutrient transporter in the intestine. By partially blocking the uptake of small protein fragments, the drug reduced cellular amino acid availability, lowered mTOR signaling, and created a metabolic state that looks surprisingly similar to protein restriction. The study was published in NPJ Aging on August 15, 2026. Gemfibrozil belongs to a class of cholesterol-lowering medicines called fibrates. Clinically, it is used mainly to lower triglycerides and alter blood lipid levels. Its traditional effects have been linked largely to PPAR-alpha, a receptor involved in fat metabolism. That is exactly why the new finding is unusual. When the researchers investigated the longevity effect, they found that it was independent of PPAR-alpha. The drug appeared to be doing something completely different from the mechanism for which it was originally developed. The key turned out to be a transporter called PEPT1, also known as SLC15A1. PEPT1 sits on cells lining the intestine and helps absorb dipeptides and tripeptides, which are tiny fragments produced when dietary protein is digested. Normally, digestive enzymes break large proteins into amino acids and small peptides. Those nutrients then cross the intestinal wall and enter the body. PEPT1 is one of the molecular doors involved in that process. The researchers found that gemfibrozil could inhibit this transporter in cells expressing PEPT1, and in isolated sections of mouse intestine, gemfibrozil reduced overall amino acid levels. In effect, nutrients were still present, but the cells experienced a weaker amino acid signal. That distinction is important because amino acids are not simply building blocks for muscle and protein. They also act as information. When amino acids are abundant, cells interpret that as a sign that nutrients are plentiful. When they become scarce, cellular behavior changes. One of the main systems reading that nutrient signal is mTOR. mTOR is a central regulator of growth and metabolism. When nutrients are abundant, mTOR helps promote protein synthesis, cell growth, and other anabolic processes. When nutrients become limited, mTOR activity falls and cells shift toward conservation, maintenance, and recycling. This pathway has attracted enormous attention in aging research because dietary restriction and several longevity interventions in laboratory animals influence mTOR signaling. Gemfibrozil lowered mTOR signaling in the researchers' cell experiments. That created a compelling possibility. Perhaps the drug was not extending healthy lifespan because it was lowering cholesterol at all. Perhaps it was partially imitating one of the molecular signals created by eating less protein. This idea fits into a much larger story in aging biology. For decades, researchers have known that reducing calories without causing malnutrition can extend lifespan in many laboratory organisms. More recently, scientists have discovered that the composition of those calories can matter almost as much as the amount. Restricting particular amino acids or lowering total protein can influence insulin signaling, mTOR, FGF21, autophagy, mitochondrial metabolism, and other systems connected with aging. But dietary restriction creates an obvious problem. A biological mechanism that looks beneficial in the laboratory may be difficult for people to maintain for years, especially in older adults who already face loss of muscle and frailty. Scientists have therefore been searching for what are sometimes called dietary restriction mimetics, interventions that reproduce selected cellular signals of nutrient restriction without requiring extreme food restriction. The new study suggests PEPT-1 could be one such point of control. Instead of changing how much protein enters the digestive tract, researchers may be able to change how strongly the intestine allows those nutrients to enter the body's metabolic signaling system. Gemfibrozil appears to interfere with that gateway. The result is not the same as fasting or protein restriction, but some of the downstream biology begins to resemble nutrient scarcity. The researchers did not stop with lifespan experiments. They also asked a more practical question. Were the animals simply surviving longer, or were they remaining healthier? For one experiment, female mice began gemfibrozil treatment at 20 months of age. That is already old for a laboratory mouse. The treatment then continued for 6 months. Researchers measured frailty using several indicators rather than relying only on survival. The treated mice showed improvement in the overall frailty phenotype, including measures involving grip strength, hearing loss, and piloerection, which is the abnormal raising or roughening of the fur seen in older or stressed mice. That detail matters because lifespan alone can be misleading. An intervention that extends survival while leaving an animal weak or chronically ill would be far less interesting than one that preserves function. Aging researchers increasingly distinguish between lifespan, the total time an organism remains alive, and health span, the portion of life spent with relatively good function. In this study, the mouse results suggest gemfibrozil may influence at least some features of health span, although the evidence is still preclinical. There was another unexpected clue from the intestine. In a mouse model of chemically induced colitis, gemfibrozil appeared to reduce disease symptoms. The authors note that this resembles effects previously associated with PEPT-1 inhibition. That does not prove the same pathway explains every benefit, but it strengthens the idea that the intestinal transporter is biologically important, rather than merely an incidental drug target. At this point, the story becomes extremely tempting to oversimplify. An old medicine extends healthy lifespan in laboratory organisms. It targets a nutrient pathway connected to protein restriction. It reduces mTOR signaling. The obvious conclusion might be that people should start taking gemfibrozil to slow aging. That conclusion is not supported by the evidence. There was no randomized human trial in this study showing that gemfibrozil slows human aging or extends human lifespan. The mechanistic work involved cells, isolated mouse intestinal tissue, laboratory model organisms, and aged mice. Even when a longevity mechanism is conserved across multiple species, translating it into humans can fail for many reasons. Human metabolism is different. Our lifespans are vastly longer. Diseases interact with aging, and a pathway that is beneficial when partially altered in an animal may produce unwanted effects when manipulated chronically in people. There is another important reason not to treat this as an anti-aging drug recommendation. Gemfibrozil is a prescription medication with meaningful contraindications and drug interactions. Official prescribing information warns against combining it with certain drugs including simvastatin and repaglinide, and it can increase the risk of muscle injury and rhabdomyolysis with some lipid-lowering combinations. It is also contraindicated in conditions including severe liver or kidney dysfunction and pre-existing gallbladder disease. This creates one of the most interesting tensions in longevity research. Repurposing an existing medicine can be much faster than developing a new drug because researchers already know a great deal about its pharmacology and clinical use. But already approved does not mean proven safe for a completely different purpose. A medication prescribed to people with severe triglyceride problems under medical supervision is not automatically appropriate for healthy people taking it for decades in an attempt to live longer. The The study itself also has an unusual scientific strength. The researchers did not simply observe that treated animals lived longer and stop there. They tried to uncover why. They moved from the drug to PEPT1 to amino acid availability and then toward nutrient-sensitive mTOR signaling. That mechanistic chain makes the finding more interesting than a simple drug screening result. The authors also reported no competing interests, although the work involved multiple institutions and was supported by research grants, including aging-related funding. The finding also raises a deeper question about modern nutrition. In aging research, more nutrients are not always better. Young organisms need abundant amino acids to grow. Older adults need adequate protein to preserve muscle. Yet chronically high nutrient signaling may also keep growth pathways such as mTOR activated when cells would sometimes benefit from switching into maintenance mode. Biology is balancing two competing demands: build enough to maintain the body, but periodically reduce growth signals enough to repair and recycle damaged components. That is why protein and aging cannot be reduced to a slogan like "eat less protein" or "eat more protein." Too little protein can contribute to muscle loss, weakness, and frailty, especially in older adults. Too much nutrient signaling may have different metabolic consequences. The optimal balance likely depends on age, activity level, disease, total energy intake, protein source, and many other factors. Gemfibrozil makes this problem especially interesting because it may separate nutrient intake from nutrient sensing. The animal can consume food, but the intestine changes how part of that protein signal reaches the body. In principle, this could allow scientists to manipulate the signal without imposing severe dietary restriction. Whether that can ever be done safely and usefully in humans remains unanswered. The next step should therefore not be people experimenting with an old cholesterol drug. The important next step is testing the mechanism more rigorously. Researchers need to know how much of the longevity effect truly depends on PEPT1, whether similar biology occurs in male and female animals across different backgrounds, what happens to muscle and other tissues during long-term treatment, and whether there is a therapeutic window where nutrient signaling falls enough to produce benefits without causing nutritional problems. Eventually, carefully designed human studies could ask whether PEPT1 modulation changes metabolic biomarkers, inflammatory pathways, frailty, or validated measures of biological aging. Only after that would it make sense to ask the much harder question of whether the intervention improves health span. For now, the most important result is not that scientists may have discovered an anti-aging pill. They have not. It is that a familiar medicine appears to have revealed a previously underappreciated connection between the intestine, amino acid absorption, nutrient sensing, and longevity. A drug developed to change the fats circulating in blood may also change the way the body interprets dietary protein. And that may be the real discovery. Sometimes the most interesting longevity mechanisms are not found by inventing an entirely new molecule. They are found when scientists discover that an old molecule has been talking to a completely different biological pathway all along.